Hello students, let's start with the new topic, that is enzyme inhibition, which is an important topic in the chapter enzyme. So enzyme inhibitor is a molecule that binds to the enzyme and decreases its activity. So it is a molecule, it's a chemical compound which binds with the enzyme and decreases its activity. So what is the function of enzyme? Enzyme binds with the substrate to form the product. Your substrate will bind with your enzyme to give you a product. Yes, any biochemical reaction, it is catalyzed with the help of enzyme. So basically this enzyme is now not available as this enzyme is binded with an inhibitor. So enzyme is not available. So your substrate will not bind with your enzyme and will not give you any product. So biochemical reaction will not occur or enzyme substrate complex will not be formed. So inhibitors are chemicals that reduces the rate of enzymatic reaction. So obviously, if there is no formation of complex, if there is no formation of product, so automatically the rate of biochemical reaction or the rate of enzymatic reaction will go on decreasing. But this chemical compounds, they just block your enzyme. They just block the enzyme, but they usually don't destroy the enzyme. They don't harm the enzyme. They don't break down your enzyme or they don't metabolize your enzyme, but only they will block, they will attach to your enzyme and make that enzyme incapable to do that biochemical reaction. So basically, what are the importance of enzyme inhibitions? So enzyme inhibitions are important for understanding the regulation of enzyme activity within the cell. So first important application of enzyme inhibition study is to determine the regulation of enzyme activity. Then to illustrate the kinetic mechanism of the enzyme catalyzing in multi-substrate reactions. So this enzyme binding to your substrate to give you a product, that is a kinetic mechanism, that is a reaction mechanism. So that reaction mechanism, if you want to study in a single reaction or in multi-substrate reaction, that enzyme inhibition can help in. Then it is useful in elucidating the cellular metabolic pathway by causing accumulation of intermediates. So if the enzyme is not available, your substrate will not bind with your enzyme. Yes, if you have inhibitors in the reaction, your inhibitors will bind with your enzyme and thus your enzyme is not available for your substrate. So substrate will not form product. So automatically your substrate concentration will go on increasing. By this type of inhibition, we can determine the metabolic pathways involved in any reactions. So next, it is used in identification of catalytic group at active site. So active site on the enzyme. So that is the site at which your enzyme, the site at which your substrate binds to the enzyme. That is nothing but active site. That is identified with the help of enzyme inhibition. Then it provides information about substrate specificity of the enzyme. So which kind of substrate can bind your enzyme can be identified with the help of enzyme inhibition. Basically, these 5 types of importance are there for enzyme inhibition. It is used in regulation of enzyme activity, then in kinetic mechanism development, then in developing metabolic pathway, then in identification of catalytic group, and in identification of substrate which can bind to the particular enzyme. This enzyme activity are of 3 types. So there are basically 3 types of enzyme inhibitors, or there are basically 3 mechanisms of enzyme inhibition: reversible, irreversible, and allosteric. So reversible inhibitors are of again 2 types, that is competitive inhibition and non-competitive inhibition, whereas irreversible inhibition is sometimes called as suicide inhibitors. And the last type is allosteric inhibition. So types of enzyme inhibitors or types of enzyme inhibition are 3: reversible, irreversible, and allosteric. We'll start one by one. Reversible inhibitor, the name itself says reversible, that is which can be reversed. Why it can be reversed? The inhibitor binds non-covalently with your enzyme and the enzyme inhibition can be reversed if the inhibitor is removed. So this is your inhibitor, this is your enzyme. Your inhibitor binds with your enzyme, forms an enzyme inhibitor complex. Now this binding of enzyme and inhibitor is non-covalent, that is a loose bond, right? So this loose bond can be broken down easily to give you enzyme and inhibitor, that is it can be reversed, and hence it is known as reversible inhibitor. Now this reversible inhibitor, the reactions which can be reversed, the complex of enzyme and inhibitors which can be broken down to give you enzyme and inhibitor, are of 2 types: competitive inhibitors and non-competitive inhibitors. Now competitive inhibitors. Competitive inhibitors are the inhibitors which closely resembles the real substrate, is regarded as the substrate analog. Now this inhibitors competes with your substrate and binds at the active site of the enzyme but does not undergo any catalysis. In competitive inhibitors, you have a competition between the inhibitor and the substrate. Why there is a competition? As your inhibitor is similar in structure to that of your substrate. Now the inhibitor and substrate have the same structure, and if it is there, it is known as substrate analog. Yes, your inhibitor is similar in structure with that of your substrate, that is substrate analog, and thus it competes for the active site on your enzyme. Yes, this is your enzyme, this is the active site on the enzyme. You have inhibitor, you have substrate. The sub-inhibitor is similar, the inhibitor and your substrate is same, right? And thus they are competing for your active site. Now this competition, who will win, depends upon two factors. The first factor is the concentration of the inhibitor and the substrate, and second factor is the affinity of the enzyme towards the inhibitor and the substrate. The first factor is the concentration of the inhibitor and the substrate. If the concentration of substrate is more, it will bind more to the enzyme. If the concentration of inhibitor is more, the inhibitor will bind more to the enzyme. So if based on the concentration of inhibitor and substrate, the winner will be decided. Whether inhibitor will bind or whether substrate will bind, that depends only on its concentration, right? The second factor deciding the winner will be affinity of your enzyme towards inhibitor and the substrate. So if the enzyme has more affinity towards the inhibitor, more binding of inhibitor to the enzyme. If the enzyme has more affinity towards the substrate, more binding of your substrate to the enzyme. So two factors affecting this competitive inhibition. First is the concentration and second is the affinity. So if we need to have a competitive inhibition, we need to see that our inhibitor have high concentration and more affinity towards the enzyme. Let's see one example. Example is succinate converting into fumarate. That is one step in your TCA cycle. The reaction is catalyzed by the enzyme succinate dehydrogenase. So for this enzyme succinate dehydrogenase, your substrate is succinate and the product is fumarate. But if I have an malonate as an inhibitor, malonate is structurally similar to your succinate. So I can call malonate as structural analog. So we can see the structure that malonate have CH2 and 2 COH group. Succinate has 2 CH2 and 2 COH groups, so only difference, but basically the structure is same. So malonate is structural analog to your succinate. Now if malonate concentration is high and if it have more affinity to succinate dehydrogenase, it will bind to succinate dehydrogenase instead of succinate. And if malonate binds to your succinate dehydrogenase, it will not form your fumarate. But if your succinate binds to your succinate dehydrogenase, it will form the product. So basically, malonate is a competitive inhibitor for succinate dehydrogenase. While studying any kind of inhibition, two factors are kept in mind: Km value and Vmax value. What is Km value? Michaelis-Menten constant, right? And what is Vmax? Vmax is the maximum velocity of enzyme activity, right? So we have already learned equation. So these two factors get affected in case of any inhibition, and that we need to represent after studying any enzyme inhibition. So in this case, in case of competitive inhibition, Km is increased. Why Km is increased? What is Km? Km is Michaelis-Menten constant, and as per equation, Km is equals to concentration of substrate. Right? But if the concentration of substrate remains as it is, yes, as your substrate is not binding with your enzyme, so you have more substrate available in your cell, right? So if your enzyme is binded with your inhibitor, your substrate will be more in concentration, and as your concentration of substrate is increasing, as it is not binding, the Km value will increase. But Vmax is unchanged, as the maximum velocity of your enzyme is not affected by this, right? So it gets unchanged. So we'll draw two figures, one using our equation and the second using our Lineweaver-Burke double reciprocal plot to show the change, that is change in Km value and change in Vmax. The second type of reversible inhibition is non-competitive inhibitor. Non-competitive inhibitor, so it's a reversible. That is, it can be reversed. Your enzyme with substrate link can be reversed. Yes, but what is the difference between competitive and non-competitive? Competitive inhibition, the inhibitors have structural analog. Here, in non-competitive, the inhibitors have no structural resemblance with the substrate. So first thing is, your inhibitor does not have any structural resemblance to your substrate. Yes. The second thing is, the inhibitor binds at the site other than the active site. In case of competitive inhibition, your inhibitor binds to the active site as it is similar to your substrate. It can easily bind to your active site. But here, there is no structural semblance of your inhibitors and your substrate. So, it doesn't bind to the active site. It binds other than the active site. Yes. And third, this binding impairs the enzyme function, and that is available in all inhibitors, right? They make your enzyme unavailable for your substrate and thus causes inhibition. Yes. Now, here, this is your enzyme. Yes. This is your inhibitor. This is your substrate. So, inhibitor and substrate don't have any structural difference, right? And your inhibitors bind to the site other than the active site. So, here, this is your active site where your substrate is going to bind, and this is the other site. Now, this is your enzyme. Yes. And your inhibitor binds to your enzyme. Here. This. Now, this enzyme conformation is different. This enzyme shape, conformation means shape. This enzyme conformation is different as compared to what this conformation is. But your substrate can identify only this. It is very difficult for substrate to identify this conformation, and hence it inhibits this conformation inhibits the binding of your substrate to form the product. Yes, we'll see this with an example. Example is silver ion, which act as an non-competitive reversible inhibition for SH group which is present on cysteine residue of any protein. So cysteine is an amino acid, right? If this amino acid is present in any protein chain, this cysteine is sulfur containing group, right? So it will have an SH group, and if this SH group binds with the silver ion, this SAG, that is SAG group will form. This conformation restrict the folding of your protein structure. So any protein molecule biosynthesis, right? Protein is first formed, long chain biosynthesis by translation, right? Now translation will give you a long chain of protein. Then that protein get folded to give quaternary structures, right? Secondary, tertiary, and then finally quaternary structure. So for this folding, if in that folding we included this non-competitive inhibitor as silver ion, it will inhibit that binding, and thus the enzyme activity will be stopped. Yes, proteins are nothing but enzymes, right? Or vice versa, I can say enzymes are nothing but protein structures. So if that protein formation, that enzyme formation is blocked with this Ag ions. Again, effect on Km and Vmax value. Km will remain unchanged, whereas Vmax will decrease. As your concentration of substrate remains unchanged, your Km value remains unchanged, whereas the Vmax, maximum velocity by which your enzyme works, get decreases. So again, we will plot two graphs, that is equation plot and Lineweaver-Burke double reciprocal plot. Then the second type of inhibition is irreversible inhibition. Irreversible inhibition, that is, it cannot be reversed. Why it cannot be reversed? As it covalently binds with a protein. Covalent binding with the protein. Thus, these inhibitors are usually toxic. As covalently, so one time binding of these inhibitors with the enzyme, so complete enzyme is blocked. Yes, example of this is iodoacetate, which is an irreversible inhibitor of the enzyme like papain and glyceraldehyde 3-phosphate. It combines with the SH group at the active site of this enzyme.